All right, the next thing that I'm going to do, um, uh, just a second. Okay, so now I'm going to go ahead and begin the process of, giving you a little bit of a rundown for what the exam is going to consist of. And in order to do that, what I want to do is share my screen. So just be kind of taking some notes on here. So your exam, over the course of the semester, we have had 5 exams. Now, the first thing that I want to say is that of all of our exams, but wait, we're there basically. EX3, EX4, EX5, and the final. So, of all of your exams, well, what we've been doing over the course of the semester is, I'm going to write in red your first shot. I'm going to call it first shot exam or first attempt. Yeah, there we go. Your first attempt score. So, exam one, let's say that you got an 80. Exam two, you got a 75. It's a hypothetical person here. Exam three, you got a 90. Exam four, you got a 92. Five, you got a 100. And then on the final, when the final rolls around, let's say you get a 96 on that. Now I'm going to switch colors. So red was your first attempt. I'm going to go with black as your second attempt. Well, the second attempt is going to be the score from the next exam. So when I'm looking at your exam one score, that's 75. Exam two, well, I'm going to bring the 90 that you got on exam three over. So boom, boom, and the 92 here, 100 here. Now, so that's our second attempt. And then the final exam for, I'm going to switch over to green. The final exam is the last attempt for each one of your exams. So I'm going to write that last attempt. And I say that it's the last attempt rather than the third attempt because if you look at exam number 5, we really only got 2 tries at that content. So 96, 96, 96, 96, 96. So that comes over here, 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 and last but not least, right there. Now, whenever I'm looking at these, I'm going to set rules to all the different parts of the gradebook, and I'm going to drop the 2 lowest grades. So what that means is that if we look at your exam one scores, the 80 and the 75 get dropped because they are lower than the 96. Exam two, the 75 and the 90 get dropped because they are lower than the 96. The 90 and the 92 for exam three get dropped. And then in this case, for exam four, well, the 92 and the 96 get dropped because that 100 is higher than those. And then last but not least, in exam five, your 100 is higher than the 96, so that gets dropped. Now, with that in mind, I think each one of these is about 12% of your grade. And this is, I believe, a standalone 15% of your grade. So that ultimately means that that 96 that you got on the final persists. That stays because that is a part of the grade. But for all these others, well, the 96 replaces a number of the other grades. So essentially what that boils down to is whenever you take your final exam, you've got a great opportunity to bring up lots of other grades. Now, that's kind of the simple rundown of the exam or the implications of the exam. The next part is, well, what's the exam going to be like? Well, one thing that I've got to pull up in a separate tab is that's no, that's not what I'm looking for. I had this up earlier. Um, and I'm just going to copy the information over. Our class here is a Tuesday, Thursday class. A Tuesday, Thursday class that meets are scheduled for 2 to 3:20 p.m. Tuesday, Thursday, 2 to 3:20. We have a designated exam time of December 12th, and that is from 2 to 4:30 p.m. So that's the designated time of the exam, and that's the time that the exam is going to be proctored. Now, in the event that you have a conflict or an issue with that, contact me. I'm going to say contact me with issues about exam time. This exam will be like all of our other exams, administered on Proctorio, administered as a Canvas quiz on Proctorio. Okay. Now, that's like the where. The next is the what. What is going to be on this exam? Well, like I said previously, we have 5 blocks. And your exam is going to be a grand total. It's going to be a little bit longer. It's going to be 55 questions. Of those 55 questions, 5 are effectively bonus. And what I mean by that is I'm going to set each question to be worth 2 points each. And so 2 times 55 equals 110. I'm going to grade this out of 100 points. So if you get 52 questions right, I'm going to say 52 times 2 is 104, and that's going to put you at a 104 out of 100. Now, when you take that into consideration, we jump back. Well, that's still true here. If you get a 104 on your exam, that 104 is higher than all of the other scores. You've got 5 questions that you could miss and still get a perfect score, still get a 100 on the exam. So this is a, built-in extra credit, a great opportunity to bring up, grab some extra points. Okay. Now, with that said, I'm going to go to our Canvas page and show you kind of the material. Close that. Okay, so now I'm going to change my share to this one. And I'm going to do the student view. Okay, so what I would recommend doing with respect to your exam is to think about it as, well, each one of these blocks with 55 total questions, there's going to be 11 questions here. And then I can scroll down and not have to rewrite that. 11 questions that pertain to exam 2, 11 questions that pertain to exam 3, 11 for 4, and 11 for 5. So if you're thinking, well, how should I prepare for this? Well, I think the best way to approach this is to look back at your old exams and say, all right, well, what were the questions that were asked over this block of material? And try to answer those questions. So I'm going to go a little bit, kind of day by day and, tackle some of those things. So here, and I'm just going to use the Canvas page as kind of my navigator of sorts. So just one second. I'm looking for something on my stop my share. I'm going to stop my share and then restart it again. Just one second. Okay. Okay, cool. Go. So I'm ready. All right. So this is my, my semester in a nutshell. What I want to do is look at my first PDF. My first PDF is, the welcome. We can scrub through that a couple of days. And why is this blank? Oh, that's not helpful. That's okay. Let me try that again. Oh, that's okay. That's right. This was our chemistry review. So I'm not. Oh, there's the marked up one. Sorry about that. So here's our marked up day where we covered all these important topics. So I would think about each one of these and say, all right, well, what am I trying to do? I'm going to go with the PowerPoint because I don't have an issue with PowerPoint. Go to the PowerPoint and think about, well, what are the subatomic particles? What's electronegativity? What's bonding? What's polarity? Here's kind of the big overview of what we talked about with respect to these. I think that doing something like this right here and knowing your subatomic particles, what they are by name, and their charges is going to be very valuable. So I would definitely make sure that I know that. Then I would go through and think about, okay, what are my major types of bonding? My major types of bonding are ionic bonding and covalent or molecular bonding. What are the differences and what are the similarities between these? Ionic bonding is where electrons are given away, whereas covalent bonding is where electrons are shared. Know those things. Now, okay, so that would be powerful. Then electronegativity. Electronegativity was a phenomenon where we talk about sharing equally and unequally electrons between atoms. So which atom wants electrons more than another in a molecule? So I'm going to clear all my drawings because that's another thing. So that's basically like three important items. So you can kind of think about that like three questions ready to go. Next up, we talked about Lipinski's rule of five, as well as we also discussed and got into, oh, rats. I'm just going to use the PowerPoints from now on. In addition to that, we talked about organic chemistry. We talked about hydrogen bonding. What is needed for hydrogen bonding? What is a hydrogen bond acceptor? What's a hydrogen bond donor? Now, when it comes to hydrogen bonding, one example that I always like to highlight is this. This is an example of hydrogen bonding. So in order for a hydrogen bond to be present, you have to have hydrogen and oxygen. Now, in this case, what we have is a hydrogen bond acceptor and a hydrogen bond donor. Our acceptor is going to be this oxygen atom. Our hydrogen bond donor is this hydrogen. So we could look at this as here's our donor. Please. This is, sorry, I shouldn't have done it that way. Okay, done. This is our donor, and this is our acceptor. Okay, so with that in mind, we have one of the important parts about bonding, hydrogen bonding. Let me clear all this out. Then we get into kind of the criteria for drugs, and that's Lipinski's rule of five. Well, there's a rule of five, not to be mistaken with, five rules. So the rule of five, or Lipinski's rules of five, rule of five, brings up, well, the number of hydrogen bond donors and the number of hydrogen bond acceptors. In addition to that, the molar or the molecular mass. So think about these. So hydrogen bonding as a great question. So we're looking at about four questions now for the exam. Then one of these rules as a fifth question. You know, remember what these rules are, how many bond donors, how many acceptors, everything like that. Now, where we can go from there is we look at a couple of different molecules. Now, this octanol water partitioning, that's not something I'm going to ask you a question about. But I'm not going to ask you what is the octanol water partition coefficient, but instead, I think that it's helpful to be able to identify, oh, well, this is good. So a range between -1 and 5. Okay, that's a fair, good question. Now, when it gets to organic chemistry, I'm going to kind of narrow your scope a little bit. We talked about the properties of carbon and the hybridization of carbon. We also talked about a couple of different organic compounds. We used organic names. So think about this as a good source for another question. Now, all of these, I would say, if I'm going to spend some time, I'm going to make sure that I know these first, let's go with five. I would say the first five examples. If a compound has one carbon only, it's meth. It has the stem of meth. Eth for two, prop for three, bute for four, pent for five. All right. So this gives us some molecule names to use. Then we have this notation, being able to translate from the condensed form to the complete structural form or the condensed form to the molecular form. These are all going to be valuable skills. Then I might ask you to look at a compound and think about, well, can you apply Lipinski's rule of five? Um, is this a good substance to consider? In which case you're applying the rules of five. Now, after that, we got into functional groups, and there's tons of functional groups. I would recommend that you do a little review of those functional groups. And one of the ways that you can review them is by looking at the questions that were given to you on exam number one, and then subsequently on exam number two. What were the questions? How extensive were those questions? I would use those as my starting point. I wouldn't necessarily dive back into the PowerPoints. So if we're making kind of a list of what's going on, um, we've got our chemistry review, we've got hydrogen bonding, we've got Lipinski's rule of five, we've got functional groups, and that's really a lot of material to wrap up. Or sorry, let me rephrase that. That's a lot of material that I think I could pretty easily put together 11 questions related to that. So I would walk through and, go through the first exam, make sure that I'm in good shape there. Next was amino acids. And we covered amino acids over a couple of days. Now, for amino acids, think about the organization of them. I'll probably ask you a question explicitly. What are the different organizations of your amino acids? There's polar positive, polar negative. There's also a third one that's polar uncharged. There's nonpolar and there's aromatics. So then go on to what makes a molecule polar or nonpolar. These are all criteria that I could ask you about. Is this compound, is this going to be, or you found a new molecule that is symmetrical? By that alone, can you classify it as polar or nonpolar? Well, if all that you know about this molecule is that it is symmetrical, then you can say that based on that alone, it is a nonpolar molecule. Because if you want it to be polar, then it's got to be non-symmetrical or asymmetrical. When it comes to nonpolar molecules, you're looking for something with no charge. So these definitions, polar and nonpolar, that relates back to, the chemistry review, but also it gives you a sense of, well, sorry, it also gives you a sense of, you see how to word this. It gives you a sense of what the, the, the amino acid is. We've got aromatic amino acids with rings, and then we've got our amino acid names. So these are all your 20 what are known as proteinogenic amino acids. These are the amino acids that are used to build proteins. These amino acids also have three-letter codes. I think that it's a fair question to ask you something as bluntly as which of the following is not an amino acid? So make sure that you know these names well enough to recognize something that's not from this list. Then a good follow-up question would be, here's four amino acids, and here are three-letter codes that are associated with them. Which one of these is not correctly assigned or correctly combined? So a question like this would, run something like this. We've got, let's say, ASP for aspartic acid. And I'm going to have ASP aspartic acid. Then we've got LEU, leucine. Then this is a U, not an N. Then we've got ISO for isoleucine. And then we've got ALA for alanine. I'm going to provide you with the question of, well, which one of these is the incorrect combination? And I would want you to look at this and say, okay, well, my my guidelines for if an amino acid is kind of correctly, or if its three-letter code is the first three letters of the amino acid. That's kind of a good starting point, but that's not always the case. That works for aspartic acid. That works for leucine. That works for alanine. But for isoleucine, that's one of your outliers. ILE is the three-letter code for isoleucine. So I want you to be able to, look at the full name and the three-letter code, see if that's correctly associated. In addition to that, there's also single letters. So which one of these? These are the one letter and three-letter codes combined with one another. So make sure that you're able to sort of separate these two and know which one's right. I put this together like this because I like to say, okay, well, I know the 26 letters of the English alphabet, and 6 of them are not used whenever I'm assigning single letters to my proteinogenic amino acids. So there we go. Now, after that, I've got the amino acids organized into your positive, negative, and uncharged groupings. Okay. That was basically day one of amino acids. So, really when it comes to our schedule, you know, amino acids, I think for this, it's going to be better part of 7 questions because one, what are the ways in which amino acids can be organized? 2, what are the correct names? Or which name is, or which of the following is not a name for an amino acid? So that's a second question. 3, be asking about 3-letter codes. 4 would be asking about 1-letter codes. 5 would be asking about kind of organization. I want you to know what amino acid is a polar positive, what amino acid is a polar negative, what amino acid is an uncharged amino acid. So that's it. I think that would be 4, maybe 5 questions right there. And so that's a lot of material just in those first 2 days. After that, we get into protein structure and protein function. So structure, this is all about organization. What types of bonds are there? And when we did this, we talked about our different levels. So being able to identify those levels, that's one question. Next up, what are the types of forces or bonds that stabilize these? For example, A peptide or a covalent bond stabilizes the primary structure. Then the secondary structure is stabilized by hydrogen bonding. Then our tertiary structure. This is simply put, it is stabilized by R group interactions. So how do the different parts of the amino acids, or the question is, I'm sorry, um, what parts or what part of a protein is stabilizing the tertiary structure? And the answer to that is R group interactions. Quaternary structure, the most important part about the quaternary structure is to know these terms right here. And a protein that has multiple different subunits is known as an oligomer or oligomer or an oligomer. And that's multiple different kind of blocks coming together to make a structure. So we can look at this and say, well, if it has two subunits, it's a dimer. If it has three, it's a trimer. Four, five, it's a tetramer or a pentamer. Now, there are also, so I'm already at probably 8 or 9 questions. Um, then what I want you to think about is what are the kind of interactions that can be formed between different parts of a protein. We've got hydrogen bonds, ionic bonds, disulfide linkages, and dispersion forces. So for these, I would highly recommend looking back at the exam that these sorts of things were asked about. This slide right here, I'll tell you right now, I'm going to ask 3 questions from this slide. This is day 9, page 7. What is on this slide is interactions between amino acid R groups. Then above that, let me clear all this out and highlight better. We've got R group interactions. And then the amino acids, single letter codes that can participate in those R group interactions. I can comfortably say there will be 3 questions coming from this. Ionic bonds. Well, it's these amino acids. Disulfide bonds. Well, it's just cysteine. Dispersion forces. Well, it's these amino acids, and those are classified as non-polar amino acids. So think about that. Be ready for that. That is day 9, protein structure, page 7. Know that slide. Okay, so yeah, the remaining slides were really just kind of a way of reflecting on and building this up. Okay, so protein function, we looked through and we learned about how proteins act. What do they do? We talked about substrate binding and active sites, basically how an enzyme grabs onto something and then acts on it. Well, here we talked about an enzyme producing a product, and we followed this kind of mechanism, this profile, where we had enzymes that added substrates in different sequences. So make sure that you're good with that. Now, that wraps up the first 2 exams content. For exam number 3, we talked about carbohydrates, nucleic acids, lipids, and protein ligand complexes. If we're going to pull 11 questions from these right here, then you can probably think, well, day 11 PDF probably has about 2, maybe 3 questions, probably 3 questions. So this is all about sugars. In addition to being about sugars, well, what makes sugars different, or what are the things that make sugars different from one another? And this ultimately went back to, sorry, this right here. Functional groups, aldehydes and ketoses. So I would definitely make sure that I know what an aldehyde is, or an aldose is, and what a ketose is. Then know these prefixes. Now I'm going to clear this out because what I'm going to do is go on to the next slide and show you some important information. So this right here, we've got our different examples. These are all aldoses, and they all have an aldehyde functional group. This is an aldehyde functional group right up here. Carbon number one is your aldehyde functional group. Now, when we talk about a sugar with three carbons, it is a triose. A sugar with four carbons is a tetrose. A sugar with five carbons is a pentose, and a sugar with six carbons is a hexose. Now, if this hexose has an aldehyde functional group, it is an aldohexose. If it has a ketone, it is a ketohexose. So I definitely know that be able to identify, looking at a sugar, what it is. Is it an aldehyde? Or does it have an aldehyde? Does it have a ketose? Or sorry, a ketone? Is it an aldose? Is it a ketose? And so on. Next up, nucleic acids. This was, well, we talked about a couple of different experiments. I've asked about those different experiments. Spoiler alert, I'm going to ask about them again. What's the Hershey-Chase experiment? What was the point of the Hershey-Chase experiment? It confirmed that DNA was the carrier of genetic information. Next up, the double helix. Well, that was credited to Maurice Wilkins, James Watson, and Francis Crick. A person who contributed substantially to all of this was Rosalind Franklin. She did the X-ray crystallography that was used to establish that structure, or the double helical structure. The Human Genome Project was published right around the year 2000, 2003-ish. And this person right here, this guy right here, J. Craig Ventnor, he's responsible for kind of coming up with an approach to solving it. He worked on a private industry approach to it. And then there was also the National Institutes of Health and I think the NSF, the National Science Foundation, that collaborated together to put together the public aspect or the public angle of it. So this was what's commonly referred to as a joint public-private venture. Now, sorry, know about these experiments, know about these people. We're looking at two questions there probably. Then DNA versus RNA. What makes them different from one another? Whenever we talk about the differences between DNA and RNA, one of the things that we're talking about and getting after is, well, what's the general structure? What are the bases of them? I would definitely know, without a doubt, what a nucleotide and what a nucleoside is. In addition to that, I would definitely know what the difference between DNA and RNA is. And I'll let you know right here. RNA is a ribonucleotide or has a sugar that is this sugar right here, ribose. Okay. The difference between ribose and deoxyribose is this right here, that hydroxide group versus this hydrogen. So those are your differences between those are the differences between a DNA and an RNA molecule. But again, whenever it comes to trying to put together a question from about nucleic acids, I would look back at the old exams. Lipids. Lipids are molecules that carry a huge amount of energy. We talked about the different classes of lipids. We also looked at how lipids differ from one another, how they pack together differently, what make them different in terms of their naming, and what a triacylglycerol is versus what a vitamin, like vitamin D. Vitamin D is a vitamin that is naturally occurring in your body, and it is a fat-soluble vitamin. Then after that, we got into cancer and chemotherapies, iron, zinc, and magnesium on exam number 4. And then on our most recent exam, we talked about vitamin C, D, and creatine. Now, I just kind of breeze through that most recent stuff because, well, it's the most recent stuff. I think what I'll do for drug design is I will, kind of take the strategy or what is the effort? What's the push? Well, you have to find a target, find a protein that is worth investigating how a drug interacts with it. Then you got to build a drug to interact with that protein. And then what you do is you go through testing. Sorry, I jumped ahead. Um, you observe how that drug is absorbed and the bioavailability of it. For instance, if that drug goes, if you ingest it in your mouth and it goes straight through your body and is excreted in your urine or something like that, well, that's not a particularly valuable drug because it's all kind of going as a waste product. That would be something that would be referred to as, got it, xenobiotic. Where is xenobiotic? There it is. A xenobiotic is a compound that's a foreign substance that your body just says, don't know what to do with that, so I'm going to get rid of it. So, in addition to that, we talked about signaling cascades. Well, know what a signaling molecule is and know what a receptor protein is. And what's that going to do? Then, these are all fantastic, or well, these two primarily are fantastic models to understand. So if I were to go ahead and or just imagine that you see this whole thing snapshotted onto a question, I'd want you to be able to, say what this is describing. This is our natural chemical. Okay, our natural chemical, what's it going to do? It's going to bind to a receptor site. Okay, then what is this right here? Well, sorry, let me clear this. If I asked what type of drug is this, if I, blocked that out and I said, okay, here's a drug. Um, what's it do? And then if I were to say the alternatively, so let me grab this again. What type of drug is this? What type of drug is this? I want you to be able to answer those sorts of questions. And now I got to clear all my markings out. And again, look at, I think that on your exam, you're asked about routes of administration. Basically, how does something get processed? If you take a drug orally, where's it going to go first? Well, it's not going to go to your bloodstream first. Instead, it's going to instead it's going to go to your gut. And then when it goes to your gut, it's going to go to your blood plasma. And then when it goes to your plasma, it's going to get processed and distributed in a number of other places. If you need something to go to your lungs first, well, inhalation is probably the best way. Know the routes of administration and kind of where the drug goes. Then we've got to get to, okay. After that, we talked about cancer and chemotherapies. Now, with cancer and chemotherapies, we talked about benign tumors as well as malignant tumors. Malignant ones are the problem ones. Benign, they could be a problem, but at the moment, they're not a problem because it's kind of confined. Whatever the growth is, whatever the problem is, it hasn't spread. A malignant tumor is one that has spread. It's distributed. It's causing problems elsewhere. And it's going to cause, well, new tumors to develop. Now, after that, we talked about tumor suppressor genes as well as normal oncogenes. Tumor suppressor genes are genes that basically will prevent cancer from developing. So your body has these mechanisms that will stop cancer from developing. And as long as there are no mutations in those tumor suppressor genes, well, you don't have to worry about tumors developing because your body's going to stop them off. If that tumor suppressor gene has a mutation, however, which is what we see right here, well, then it's not going to be able to function normally. It's not going to be able to stop what it needs to stop, in which case cancer can develop. Then, let's see, we talked about cancer therapies. There's so many different approaches to resolving and stopping cancer. What are they? Well, surgery is one approach, radiation, and many cases or many times whenever someone has a cancer diagnosis, it's throw everything at it. Um, throw the kitchen sink at it, do the targeted therapy, the stem cell therapy, hormone therapy, all of the different things. Then one question that came up a couple of times was what is cisplatin and what is an alkylating agent? Then there was questions about hormonal therapies. If your body is producing hormones that cause a cancer to develop, well then maybe you can block that hormone, in which case maybe that's going to stop the cancer from developing. Then we talked about iron as a dietary supplement and what it's good for. Iron's used in tons and tons of different biochemical reactions within your body. Very importantly, it's essential for the transport of oxygen throughout your body. It's also involved in numerous different metabolic processes. And in the event that you're experiencing iron deficiency, well, then you might be experiencing fatigue or weakness or, poor circulation. Maybe parts of your body turn slightly blue or they're very cold. That could be a sign that you're not getting enough iron in your diet, in which case you might go on to take or either change your diet by adding things like eggs or meat or leafy green vegetables like broccoli or kale or pumpkin seeds. And so that's one approach to do it. And that can help you rebound from that. There are recommended daily allowances or amounts of iron that you need. And then there's also upper limits. Don't want to take too much because that can cause a problem. Now, we also talked about magnesium and zinc. Well, magnesium is used in tons of different biochemical reactions. It stabilizes ATP, your body's energy currency, and it's also involved in helping regulate calcium flow into nerve cells as well as into muscle cells. And we talked about different different supplemental forms of magnesium as well as zinc. Zinc is involved in immune function and DNA synthesis, and zinc is something that basically helps stabilize DNA. After that, vitamin C. This is a water-soluble vitamin. Vitamin C is involved in and found in tons of different fruits. It's in your citrus fruits as well as in leafy greens. Um, and one thing that it helps do is it basically helps your body prevent oxidation. And so in addition to that, it helps fibrous proteins. So vitamin C is involved in, wound healing. And what it helps do is to make or to take the amino acid proline and modify it to make a 4-hydroxyproline as well as a 3-hydroxyproline. So that's a form or a variation of proline that's pretty essential for um, collagen formation. So it helps with wound healing. In addition to that, um, vitamin C helps your body maintain the proper composition of iron. So then we've got vitamin D, which we took a little bit of a deeper dive into. Rather than just talking about it as a lipid or a fat-soluble vitamin, we talked about vitamin D. Give this a second to load. Okay, there we go. Vitamin D and some of the benefits that it has. It is involved in helping bone diseases, specifically the rickets. Vitamin D is processed by UV light. So when you go outside and there's UV light exposure that you get, well, that actually kind of helps you out. Too much UV light can be cancerous, but kind of the right amount is good for you. So one thing that I do want you to know and be able to answer is, well, what organs are essential for vitamin D production? Well, you've got your liver and your kidney, and your liver is going to help with that processing. And vitamin D is going to be processed by going to your liver first and then to your kidneys. Um, then we just kind of talked about the different roles that vitamin D plays in expression of different genes. And there's a potential that vitamin D can have a positive benefit to cardiovascular health as well as cancer. Then last but not least, we talked about creatine. Creatine is a very commonly taken dietary supplement. It's made from a couple of different amino acids, cysteine, glycine, and arginine. I would know those amino acids. What does it help with? Well, it helps you kind of it helps you understand or it helps your body kind of augment or supplement your ATP stores and make sure that you have enough so that, whenever your ATP drops down, well, there's creatine there to kind of supplement. Where does creatine come from? Meats, typically. Not exclusively, but typically it's going to come from meats. And in part because your body can produce creatine. Your liver, the kidneys, and your pancreas all contribute to the production of creatine. And they all work together to put creatine into any cells that you have that need it that make this phosphocreatine system. And that phosphocreatine system basically supplements whenever your ATP stores are low. So those are kind of the high points of creatine. Again, what I would recommend that you do is look over the exams and look at the questions that were posed there to see if there's something that's like, oh, okay, that's I got that one right. Great. Look at those questions, and that's going to put you in a good place for the final. Like I said, the final is Thursday, December 12th from 2 p.m. to 4:30 p.m. In the event that you need to reschedule or something like that, just contact me. If you want to take it early, I can help you out with that. Um, but again, email me, joelgray@txstate.edu, and we'll get it taken care of. But with that said, I'm going to go ahead and stop my recording. And I'm going to say, I hope that you enjoyed this class. Thanks for being in my class. I hope you do well on this final as well as all of your other finals. And I will see you later.